Hi everyone, thank you for attending the sample prep session. My name is Elsa Molina, I'm from the Salk Institute and I will be the chair today for this session. So we will start with our sponsor, Takara. Thank you very much for that.
Thank you, Molina. Hello everyone, my name is Shuwen Chen. I'm the Senior Product Manager for single-cell NGS automation at Takara Bio. And today it is a great pleasure for me to introduce our latest solutions for automated high-throughput single-cell total RNA or WGA sequencing. So first, I would like to start with a very quick and brief overview of Takara Bio. We offer reagents and kits that cover the gamut of cell and molecular biology from NGS, PCR, cloning, to gene delivery and functional genomics. Indeed, we offer our reagents and kits through OEM partnerships to MDX companies and tool providers who rely on the quality and high performance of our products to power their own kits. As you may know, our companies used to be quite well known for the template switching-based smarter technology for single-cell RNA-seq, which has full gene body coverage and high gene detection sensitivity. Combined, this technology offered considerable advantage over 3' end counting methods to detect SNPs and alternative splicing isoforms. However, although our single-cell RNA-seq or single-cell DNA-seq technologies have been featured in many publications, before, the throughput of these products have not yet met customers' need. And if you look at those single-cell NGS technologies available on the market, there are a few high-throughput technologies available. However, almost all of these high-throughput technologies require customers to make some trade-off or compromise between achieving high cell throughput and obtaining ultra-sensitive data. And now we are very pleased to let you know that with the Shasta single-cell system and the new Shasta NGS chemistries, customers will no longer have to make such compromise. Last year, we announced the launch of our new single-cell system, Shasta, together with two new NGS chemistries, Shasta Total RNA-seq for high-throughput single-cell total RNA-seq with full gene body coverage, and Shasta Whole Genome Amplification Kit, or Shasta WGA, for customers to perform high-throughput single-cell DNA-seq with whole genome coverage. So let's get started with Shasta Total RNA-seq. As I mentioned, it is a high-throughput technology for you to perform single-cell RNA-seq with full gene body coverage. With this technology, you can get up to 100,000 cells spanning up to 12 different samples per experiment. And because the chemistry comes with full gene body coverage, you can leverage the data to detect alternative splicing isoforms and gene fusions. And what makes this chemistry very unique is that it deploys a random priming-based reverse transcription, so you not only get sequence information for mRNA, you can also get readout for those non-polyadenylated RNAs, such as lincRNAs. So to demonstrate the power of this method, we generated some data in collaboration with Dr. Ting Wang from Washington University using isogenic p53 knockout and the wild-type counterpart, either alone or exposed to some epitherapy treatment. And looking at basic sequencing metrics, from this experiment, we obtained over 11,000 single cells spanning the 4 different sample groups, and we detected about 10,000 genes and about 40,000 transcripts per single cell for all the different samples used in this experiment. So we think this sensitivity was pretty good, and Dr. Ting Wang was pretty happy with it. And more than that, not only are we able to capture those protein coding genes, because of the random priming-based RT, we were also able to detect thousands of lincRNAs per single cell. And on top of that, we can also perform UMAP clustering based on the lincRNA expression patterns of these individual cells. And with the new Cogent tools for bioinformatics analysis, we can also identify those differentially expressed coding or non-coding genes among different sample groups. And besides this, our R&D scientist has also conducted some extra experiments to see how we can demonstrate the benefit of full gene body coverage. And we paid attention to this gene, GATA1, which was known to be an important transcription factor in K562 and hematopoietic lineage in general. So in this browser shot, the read coverage of the GATA1 transcript was shown in these blue and red signal tracks, and loops connecting exons indicated detected splice junctions. As you can tell, in these blue signal tracks representative of Shasta total RNA-seq, reads were relatively evenly distributed along the GATA1 transcript, and all the non-splice junctions for this gene were successfully detected. We even detected a normal or a minor exon skipping event passing over exon 2. While on the other hand, if we look at those red signal tracks representative of a major single-cell competitor, all the splice junctions detected were heavily biased against the 3' end, and those splice junctions on the 5' end were completely missed. Besides that, when we compared the number of splice junctions detected based on the total number of annotated or non-splice junctions that we have previously found in K562 cells, we found that Shasta total RNA-seq detected more splice junctions than the major single-cell competitor. And when we normalized those annotated and non-splice junctions against the gene body, we found that Shasta total RNA-seq more evenly detected splice junctions across the gene body, while with the major competitor's method, all the splice junctions detected were biased or enriched at the transcript ends. Besides that, we also found Shasta total RNA-seq detected more splice junctions than the major competitor at different read thresholds. At the read threshold of 10, Shasta total RNA-seq detected twice as many splice junctions as compared to the competitor. So next, let's pay attention to another high-throughput technology we developed recently, which is Shasta WGA. As I mentioned, this technology allows you to get high-throughput single-cell DNA-seq libraries with whole genome coverage for up to 1,500 single cells per experiment. And this technology was powered on the Shasta single-cell system and our Picoplex WGA chemistry. And with this chemistry, you will be able to get single-cell DNA-seq libraries, and at the same time, you can get CNV and SNV information at single-cell resolution with very low sequencing depths. So one of our early access customers, Dr. Ilaria from St. Jude Children's Hospital, used this Shasta WGA technology to process the CD3 positive T cells and the leukemic blast cells from the bone marrow of 2 B-cell leukemia patients, S1 and S2. And using Shasta WGA, she successfully detected segmental gain on chromosome 1q at different copy number levels, and at the same time, within the leukemic blast cells of patient S1, she detected a biologically interesting small subclone with concurrent copy number loss on chromosome 9p and copy number gain on chromosome 9q. And for this experiment, as you can see on the lower right, she processed about 1,300 cells in total. And more than that, with Shasta WGA, we now give customers the ability to perform UMAP clustering from your single-cell DNA-seq data for these individual cells based on their copy number profiles. And in this way, although each single cell only gets, for example, 250,000 to or about 500,000 reads per single cell, which is too shallow for SNV analysis. By clustering the single cells together in each individual cluster, we will get sufficient sequencing depths for SNV analysis. And using this pseudo-bulk approach, we successfully detected somatic SNV within the Plur gene in the leukemic blast cells of patient S2. And as you can see on this UMAP plot on the left-hand side and the IGV browser shot on the right-hand side, this somatic SNV of the Plur gene was only present in the leukemic blast cells of patient S2. It was not present in the leukemia cells of the other patient. It was also not present in the normal CD3 positive T cells. Although we don't know about the biological significance of this SNV within the Plur gene yet, the ability of Shasta WGA to capture these CNVs and SNVs at single-cell resolution with very low sequencing depths makes it a perfect tool for those oncology researchers who would like to discover biomarkers for personalized or precision medicine. So this wraps up the technical part of my presentation. This is a very brief talk, but I hope with the data that I have covered so far, now you know that we launched this Shasta single-cell system together with two very cool high-throughput technologies for you to conduct single-cell total RNA-seq and single-cell DNA-seq with full gene body coverage or whole genome coverage so that you can retain coverage and sensitivity at scale without making any compromise on your data quality. And because the workflows have been automated on the Shasta single-cell system, they are very easy to carry out. And at the beginning of 2025, we launched the free Cogent tools for bioinformatics analysis. So we also make the data analysis very streamlined for these technologies. So we hope all these different tools can help you discover more biomarkers and make more discoveries and breakthroughs. So thank you very much. With that, that's good science. Please let me know if you have any questions.
Thank you very much. So actually for the sponsors, the question will be at the end. Yes. So now I'm going to introduce you to Dr. Mandovi Chatterjee. She's the director of the single-cell core at the Harvard Medical School, and she's going to present you, of course, all the best tips for sample prep for single-cell RNA-seq. Thank you very much.
Thanks, Elsa. Hi, everyone. I'm Mandovi Chatterjee. I'm the director of the single-cell core at Harvard Medical School. And today I'm going to share some practical guidelines for sample preparation for single-cell RNA sequencing. Briefly about our core, we are a small team supported by an incredible group of faculty advisors. Our user base includes all the academic and industry labs in the greater Boston area, mainly. We also offer our services to labs in other parts of the United States and overseas as well. Since our inception in 2016, we have worked on a plethora of tissue types across many different organisms. We support a whole suite of high-throughput single-cell and spatial technologies. I encourage you to visit our website for more information if you're interested. So today, I'm going to give you a brief overview of the field. I'll mainly focus on the sample prep and its importance. I will discuss different approaches for sample prep and common challenges and solution. So the first single-cell RNA sequencing paper was published in 2009, where 5,000 or so genes were detected from blastomeres compared to the microarray data, and some novel splice variants were detected as well. Since then, pretty much every year, a paper or a method paper has been published with some incremental yet very important advancement. For example, CellSeq first introduced the concept of barcoding, which enables sample pooling. MarsSeq introduced the concept of UMI, or unique molecular identifier, for accurate RNA quantification, and so on. However, up until 2015, all of these technologies were plate-based. They could be scaled up or automatized, but they were labor-intensive. In 2015, two droplet-based technologies were published, DropSeq and InDrops, which resulted in a paradigm shift. They enabled profiling of tens of thousands of cells in a cost-effective manner. Advancement in next-generation sequencing also complemented this effort. Following this, there were several other high-throughput single-cell RNA sequencing technologies were published over the years. And gradually, the focus of the field shifted towards multi-omic analysis, where people were interested in looking at multiple modalities from single cell. And now, I hope like from the exhibit hall itself, you can tell that spatial transcriptomics is the thing. There are several commercial players at the moment. I have hands-on experience for some of them, not all of them. So based on my experience, I can sort of divide them into these three broad categories, and there are many more, I know. So the droplet-based method, where single cell is encapsulated within nanoliter-sized droplet along with the barcoding bead. Within the droplet, the cells are lysed, their RNA content are captured and barcoded by reverse transcription. In nano or microwell-based methods, single-cell suspension and the barcoding bead are sequentially loaded in tiny wells, and within these nano or microwells, the cells are lysed and their mRNA content are captured. While in split pooling-based methods, the barcoding is achieved by either combinatorial barcoding or combinatorial indexing through multiple rounds of splitting and pooling. There are many technical differences between these technologies. However, the overall workflow for all of these technologies remain the same, where you need to prepare very high-quality sample first, then you can barcode the RNA content using your preferred technology. And then the barcoded RNA is subjected to library prep, where the RNA is made compatible for sequencing using one of the next-gen sequencing platforms. And finally, the data gets analyzed. Over the years, it has become very clear to us that sample prep is one of the most critical steps in this overall workflow. You may have heard this phrase several times, garbage in and garbage out. So if your sample input is of poor quality, there is no way you're going to get good quality data. So therefore, for the rest of my talk today, I'm going to focus on this very critical part of the workflow, that is sample prep. I'm going to talk about what high quality sample means, how to achieve them, and I'll share some of the practical guidelines based on my experience, and I'll discuss some popular approaches. So what do I mean by high sample quality? 90% or more viability for your sample is a must. The cells need to be intact up until the point they are barcoded. The cells should not clump. If they clump, it is not a single cell experiment anymore, and little or no debris is recommended. So what happens if your sample has low viability? So cells lyse when they die, and they release their mRNA content in the sample, which increases the background. And this ambient free-floating RNA is depicted by this green color in this cartoon. They get barcoded along with the RNA coming out of intact cells. And so higher the ambient free-floating RNA, higher is the background in your sample, which is depicted by this plot on your right. And if your sample is dominated by this free-floating RNA, you will end up wasting a lot of sequencing reads because they're going to get eliminated eventually from the analysis. Transcriptome is stress-sensitive as well. So this group looked at satellite cells from skeletal muscle, and they wanted to look at the effect of dissociation on these cells, on gene expression mainly. So by single molecule FISH, they looked at the expression of one of the well-established stress response genes, FOS, and they noticed that FOS expression was significantly higher post-dissociation. They went ahead and looked at the slide didn't come out right. Anyway, so they went ahead and looked at the effect of the duration of dissociation on these cells, and they noticed that there was a stark difference between the cell cluster distribution and gene expression pattern between two conditions that they tested, short and long dissociation time. So based on what I have told you so far, I hope you appreciate the importance of sample prep for single-cell RNA sequencing. So how can you achieve them? So based on your tissue type, you may or may not need to dissociate your samples. Unfortunately, there is no universal sample prep protocol. You will have to choose the appropriate dissociation enzyme. Then you'll have to optimize their concentration and duration of their treatment. And dissociation has a huge impact on cell viability and the quality of the single-cell suspension. So for most of the tissue types, once you dissociate, you end up with a lot of debris. There are dead cells present. There are clumps. So a lot of people prefer to use dead cell removal kit. And to remove the debris and the cell clumps, often density gradient or simply filtering helps. You may or may not need to enrich your cell type of interest, either by MAX or FAX. I recommend cell enrichment only if it is necessary and using broader cell surface marker instead of stringent sorting conditions. Negative sorting can be used as well. A lot of people who choose to perform FAX sorting, they often include dead cell stain to exclude them eventually. And finally, you need to check the quality of your cells, of your sample. So what do you look at? So you look at the cell concentration, you look at the quality of the single cell suspension, and you check the viability. So as I mentioned, your cell type of interest can be enriched by either FAX or MAX, a fluorescence-activated cell sorting or magnetic-activated cell sorting. They both have certain advantages and disadvantages. So enrichment by FAX is very robust and can be really useful for rare population of cells. It yields good single-cell suspension, and people often add a dead cell stain to exclude the dead cells from the samples. However, FAX exerts high pressure to sort the cells, which can create a lot of stress situation for the cells, and that can affect the transcriptome. It can introduce bias in your experiment as well if a very stringent condition is used. And finally, it adds time to your sample prep protocol. On the other hand, MAX is gentle on your sample. The protocol is fast. You can process multiple samples parallelly using MAX, and it is not limited by FAX sorter availability. However, number of antibodies conjugated with magnetic bead is limited, and also enrichment by MAX is not precise, so it does not work for rare population. We still rely mostly on freshly prepared single-cell prep because of the high data quality. However, it is time-consuming. Therefore, it limits the number of samples you can process at a time, and achieving high cell viability is often challenging for many tissue types. Thankfully, we have some alternative approaches available out there. Single nuclei preparation has become pretty popular. And the protocol is fast. It allows you to accommodate complex experimental designs involving many samples. Basically, you need to harvest your tissue of interest, and you fresh freeze them, and you can store them at -80 for a long period of time. However, the sample prep is challenging. Nuclei are leaky structure, therefore you need to be very gentle when you prep them. You need to be fast. You need to maintain cold chain all through the process. Nuclei prep often leads to a lot of debris in the cells. It's sample dependent though. Because of their small size, counting can be a challenge as well. And since single-cell RNA sequencing is basically a number game, miscounting in any direction is a problem. And it's not compatible for all cell population. It works great for neurons, adipocytes. However, if your interest lies in immune cell population, nuclei prep is not something I would recommend. Finally, fixing samples is also gaining some tractions. It simplifies experimental design involving many samples. It has enabled us to profile biobank samples as well. There are multiple fixatives available out there, but the choice of fixative depends on the experimental goal. One disadvantage I can think of is while fixing the cells, the cell loss is pretty high. Therefore, for small sample input, I would probably not go for fixed samples. So with that, I would end my talk. I hope you now appreciate the importance of sample prep. And if any one of you is planning to do a single-cell RNA sequencing, I hope you have some information to where to start. So with that, I would like to thank all of you for your attention, and I'll be happy to take any questions. No questions. I guess I did a great job. Yeah, sure.
Do you have any recommendation for different fixation, let's say for different assays like single-cell RNA-seq, single-cell DNA-seq, or ATAC? Is there, like ATAC, probably people don't use fixation that much, but do you have any recommendations?
So, like I said, for fixation, what kind of fixative you will use depends on the biological question. For example, paraformaldehyde, for example, that does a very good job of protecting the cellular architecture and the protein, but then the RNA integrity is not very well maintained. So if somebody is planning to do a parallel profiling of protein, that would be the way to go, but the single-cell RNA sequencing part has to be probe-based, like what 10x does. Or DSP is there, which can maintain both protein and RNA integrity, but it's slightly tricky how you rehydrate the sample.
Thank you. Yeah.
Yeah, great. Thanks. A very nice overview. I was wondering if I could follow up a little bit on your observation that max is not appropriate for rare cells. Is that specificity? Is it sensitivity? Is it a combination of those problems?
Specificity. So it can handle a lot of cells, but the sorting is not very precise. Mm-hmm. So there can be a lot of, you may end up with some cells that you're actually not planning to sort, but they're present in your sample, if that makes sense.
So I guess that's a matter more of, you're going to do profiling after that, right? So if you think of it as an enrichment, right, you may still be able to sort.
Yes, you still be able to sort, but then you cannot. It's hard when your focus is to isolate a rare population, mm-hmm, because it's a very small number in your entire sample. So now, if you are trying to enrich a smaller subpopulation using MAX, you may end up with a lot more non-specific cell in your sample, and that might be an issue.
So I guess depending on your question, it may be okay, or it
absolutely
may be confounding.
Absolutely.
Okay, thanks.
Thank you.
Hi, thank you for your talk. I work in a very small genomics core, small university. We don't have a ton of people doing single cell, and the way that we're set up, we have individual labs and investigators doing their own sample prep and then handing their cells off to me to process. Mm-hmm. And I have a really hard time convincing investigators that they need to optimize their cell prep. Do you have any tips or recommendation for how to stress that to them?
Sure. First, well, I'm sorry.
Thank you.
First of all, I usually meet with all of these investigators before I even start plan an experiment, and probably you do that as well. If it helps, do show them all the data. And another thing we do is that, we do consult every single user, but then at the end, we don't have control of what they're bringing to us. But once they bring the samples to us, the first thing that we do is we count. We have them wait until we are done counting. We look at the viability, and we tell them that if the viability is less than 70%, I'm not going to run.
Okay, awesome.
Because
Thank
I know
you.
that many months down the road, if the data looks bad, they're going to come and ask me, what did you do wrong?
Yeah, that's a problem I see too. And when we started out with single cell, I was doing the cell counting for people, but then I realized that some people weren't even looking at their cells before they were handing them off to me.
So I would set some ground rules and do refuse samples if you can.
Awesome. Thank you.
Sure.
Yeah, core people do magic things sometimes. But yeah, like as Mandovi told us, it's very, very important, your sample prep, so you need to emphasize that to the user and maybe like write that down on your board or make it clear, you know, even when you send the email to the user before they meet with you, tell them that it's very important.
Because it's your reputation that's at stake.
Yeah,
So, yeah.
Okay, hello. I have a similar question. This just happened last week where somebody didn't count before fixing and then was convinced that fixing is why everything was clumpy. So how do you, so then I had to go through the whole thing to explain, well, you need to look at things before you do anything, and then once you do something, you have to look at them again, and then look at them again. So I would think people would know this, but they don't.
So one of the things I always suggest that the day of your experiment should not be the first time. They should be
Absolutely. Yeah, this is just a test count,
yeah,
so that's good.
prepping this. Yeah, so fixation often leads to cell clumpings, but filtration can help to get rid of the clumps. But that's one of the reasons where the cell loss is higher in fixation. There is no, I can't think of any way you can make the cells not clump after fixation. But yeah, if you have to run those samples, it's possible that people have tried several times and that's the best they can do. But then I would give them a good, like in writing, I would tell them that I'm not liable for the good data quality after this. So they should know what to expect. So my understanding is that setting the expectation is really important.
Thank you.
Sure.
Thank you. So last quick question, please.
Hi, I'm Mike from University of Alberta. I run a genomics core as well. And kind of a similarly small, people hand us their samples. And more of a comment, I guess, is we actually have like these very elaborate like paperwork documents now that we'll sign off on viability when they're there. Because again, we can't always refuse samples because they're old mice or they're transplants.
Do people read them?
So we force them to fill them out now.
Okay.
And show up and sign them and say if their viability is under X amount, like we're not responsible for this. So when they come back in like a year, when they're looking at their data, we can now tell them like you signed this, you accepted the thing. So more of just like an operational thing for people. That's what we kind of do. So thanks. But
Yeah.
good talk though.
Thanks. Thank you very much.
Thank you very much. Thank you for the talk, and it was very important, I think, to remind people how sample prep is important. Okay, thank you everyone. So for people who are not here earlier today, so my name is Elsa Molina. I'm leading the single cell and spatial omics core at the Salk Institute. And now we are going to talk about a very unusual and exciting project at the same time. So this project is to study the effects of microgravity on astronauts, especially its aging effect. So for the past 2 years, the single cell and spatial omics core have been collaborating with a lab at the UC San Diego, as well as with Axiom Space and the International Space Station to explore the effect of spaceflight on human hematopoietic stem cells. So Christian from my team and myself today, we are going to present you how we handle the samples and how we successfully perform single-cell RNA-seq on astronauts' blood stem cells. So before, we would like to thank very much Dr. Arpita Kulkarni, who unfortunately cannot be with us today, to give us the occasion to present you this project. So let's talk a little bit about the Salk Institute. So it was established in 1960 by Dr. Jonas Salk, who was the developer of the first safe and effective polio vaccine. The Salk Institute is also known for its architecture. It was designed by Louis Kahn. So the single-cell and spatial omics core is actually a new core from July 2024, and it was formerly known as the next-generation sequencing core. So when we performed this project on astronauts, we were providing short and long-read sequencing, bulk and low input library prep, single-cell, single-nuclei library prep, ATAC-seq library prep, but as well as some spatial transcriptomics assay and digital gene expression assay. So now let's move to the actual project. So the Space Hematopoietic Stem Cell Aging Project, also known as SASHA, as I previously said, it was a collaboration with the laboratory of Dr. Catriona Jamison at UCSD, Sanford Stem Cell Institute. And Dr. Jamison has sent several projects into space and has been collaborating a lot with the International Space Station and to basically make some discovery about cancer and to improve if possible, and deliver novel therapeutics to patients. So the aim of the SASHA project is to study how microgravity affects stem cells, aging, and cancer. It was an ISS-sponsored study, and we worked on two Axiom space missions, IX2 in 2023 and IX3 mission in 2024. So for your information, the woman that you see on IX2, so on the top right corner, her name is Rayana, and she is the first laboratory technician that was trained to go to space. So you see, you never know, maybe you one day on one of your technicians will go to space to perform experiments. So now let's move to the experimental design, and Christian is going to present you how he did, because he was the main person in a lab who performed the assay.
Hello guys, like Elsa said, I was one that was part of the team that was in charge of the assay for 10x Genomics. And for the experiment design, there was 4 astronauts that were involved on the AX2 mission, and we had to collect their peripheral blood. The mission started on May 21st, 2023, and there was 5 time points that were important for us to collect their blood. The first time point was 1 day before launch, and the second time point was 10 days in flight. The third time point was 1 day after returning to Earth, and then 30 days after returning, and then 1 year after returning. And for each sample, we had to do 2 cell fractions, and the first fraction was with PBMC, and the second fraction was with hematopoietic stem cells and progenitor cells. For the assay, there was a couple of steps that we had to do, and the first step was isolating the cells, but the isolation of the cells was actually done by the Sensor Consortium team at UCSD. My role was to actually perform the 10x assay, which started with encapsulating the cells in the gem, and then creating cDNA and library prep for the samples. We had to do a custom pooling because we had to prioritize the stem cells, which we did deep sequencing for these samples. And we used an NovaSeq 6000 to perform this sequencing, and the data analysis was actually done by the UCSD team as well. And I'll hand it back to Dr. Molina, where she will go in detail of all we did.
Thank you. So now I'm going to discuss, of course, the challenges that we faced. So I'm going to list those challenges and explain you how we resolve them. So the first challenge was actually the mission itself. We didn't know if on that day in May the rocket will be launched or not. Okay, and so if astronauts are sick or if the weather is not good for the rocket to be launched, I mean, everything can be actually modified. The whole mission is, the timeline, everything is modified. So we cannot really actually have hands on this challenge. We just need to wait and see what's going to happen. The second challenge is the timeline. So the timeline is everything. So as Christian showed you, we had to collect specific days before the launch and after the launch. So of course, if the launch was moved back or delayed, it changes everything. So as a core facilities, it means that you have to be very flexible if you want to work on this type of project. It was on-call work, and we had to work during weekends and also during the night to perform this project. I don't know if you have noticed, but the Axiom 2 mission and Axiom 3 mission are actually over 1 year. So it has been 2 years that we actually expect to work on the weekends or after hours for this big project. And now let's talk about cell viability. As we discussed in a previous presentation, it's kind of a challenge with samples collected actually in Florida or even in space. So we did our best. So basically, when samples were arriving in San Diego, we worked very fast to process them, to process with the first step of the 10x Genomics assay. And we were also at the same time very gentle with the cells. And now about quantity. So you can imagine quantity is also a challenge when samples are collected on the East Coast or in the flight, and they have traveled to Earth to be processed. So what we did is actually, for some of the samples, of course, we didn't have enough quantity of stem cells because the most principal fraction that we were supposed to study were the stem cells. So when we didn't have enough stem cells to proceed with step 1, we actually pool fraction together. So in one samples, we had stem cells and also the PBMC. So for those samples where we had a mix, we had 2 fractions, we actually made a custom sequencing pool to be able to attribute enough reads on the stem cells and not actually on the other cells in the pool to be able to see the gene of interest that we wanted to see in the data analysis. And guess what? We successfully sequenced actually more than 80 libraries in total. And we were able to see all the genes that we wanted to see within the stem cells for this project. So it was a very, very challenging project, as you can imagine. This big project resulted in 2 papers currently under revision, and unfortunately I cannot disclose any data yet. So hopefully you will be able to see the data online soon. But this is a very important project that after that now we know how to proceed with samples that are very sensitive, and definitely we didn't have 70% viability when we did the 10x genomics assay, but we were able to overcome those issues and be able to have a great data at the end. And so for that, so I would like to thank, of course, the team at the UC San Diego Center for Stem Cell Institute, as well as my team, everyone who worked on those samples, because I know they were pretty stressed because they could not make mistakes. We couldn't redo it again. And we didn't make any mistakes, so everything went well. As well as the team at UC San Diego who did the data analysis, and of course, people from Axiom Space. Thank you very much. Okay, so we have time for questions.
I have a question.
Okay.
So on the experimental design, so this is a time course experiment, which is always hard to sort of design when you're handling a fresh sample. So you handle fresh samples?
Correct.
And I guess you process them at different times.
Correct.
Did you see any batch effect?
So this is the person with the team at the UC San Diego who is doing the data analysis, and that I cannot tell you right now. Yes?
Again, this may fall into kind of another like non-disclosure issue, but with those samples that were collected in space at that time, was that something you would have to like wait until like a ship would come back from space?
Exactly.
Okay.
So actually the samples that was collected in space, this is the first time that samples were collected in space. So we are actually following the twin study. If you know it, when there was actually one twin that went into space and one to stay on Earth. But at that time, samples were collecting before the launch and after, when they were back on Earth. So it's the first time that samples are collecting into space, and they are studied on Earth. So the blood was collected just before they were ready to come back. Okay, so they stayed 10 days, and it was basically they collected the blood on day 10, and then when astronauts landed, you can say that on Earth, there was a private helicopter who carried the samples from, because I think they arrived in Gulf of Mexico, and carried the samples directly to San Diego, where the team processed the blood.
Amazing, thank you.
You're welcome.
Hello,
Hi.
kind of following on the space samples.
Yes.
How are those samples protected from stress? Because single-cell sequencing is stress-sensitive, as we have emphasized before. I was just curious about, re-entry into Earth and all that. And I don't know if you can even disclose any of
yes.
that.
Yes. So we noticed that some samples, at the end with the data analysis, some samples, we didn't see any stress marker in them. And we believe, if I can come back, because there are one of the astronauts was actually a pretty experienced private astronaut. And this is the commander, Peggy Wilson. So the woman on the top left. So we present you AX2 mission, but we did approximately the same design for AX3 mission. So we know that we have a control, so we know that actually the samples were not really stressed by the travel from space. I think it's more, if they were stressed, cells were dead anyway, so we did not process those cells.
Right.
Okay? So the stress markers that we are seeing, it's more like epigenetics, and are related to the stress of the astronauts themselves.
Okay. Oh,
Yeah.
Perfect. Thank you so much.
No problem.
Hello.
Hi. Oh, sorry.
Thanks for sharing this amazing project. So I'm kind of surprised you guys didn't use the fixed cell method.
Okay, can you please repeat? Sorry.
The fixed RNA platform method with the 10X?
We didn't fix this.
You didn't fix the cells at all.
Okay, so that it would be more a question for the UCSD team who prepped the cells. I'm not really quite sure why they didn't fix the cells.
So like in the space, they didn't fix
Oh,
the
no,
cell?
no, 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 yes, no.
So,
no.
I mean, like, I'm wondering what you're going to see in the data because, you know, the gravity issue,
Yes.
all those things, you know,
Yes.
when it's traveled to the,
Yes,
you know,
yes.
from space to Earth, it's
Yeah.
going to change the gene
So
expression a
there are
lot.
some info that were not disclosed to me too, you know?
Yeah, of course, of course. But
I
like
just-
the basic
yes.
concept about this, so even I think to fix the cell in the space would be really great to fix that gene expression, that status. So,
Yes,
yeah.
yes.
Okay.
I know I suggested actually to the NASA team that was working on that, I suggested like a product to preserve the RNA, but I'm not sure if they used it or not.
Okay, thank you.
Hi.
Hi.
I wanted to ask whether the cell-free fraction of the blood has been analyzed as well.
Had enough, sorry.
Whether the cell-free fraction, cell-free RNA, for example, has been analyzed as well, sequence.
So we analyzed only the fraction stem cells and PBMC. That's
Okay.
it.
Thank you.
Okay, thank you very much, everyone. So now we are going to finish with 10x Genomics. Thank you.
Great. Well, I just wanted to start off by thanking the organizers for ABRF, as well as the organizers for this session, as well as Arpita Kulkarni, who invited us to participate today. But my name is Daphne Cooper. I'm a science and technology advisor with 10x Genomics. I started back in 2018, and throughout the past 7 years have helped many different researchers and cores with their sample prep workflows for 10x Genomics. So we're going to talk about that today. And so you've learned from our previous speaker and presentation from Elsa about some really exciting applications of single cell. And with single cell, of course, you're able to resolve through these very complex biological systems or tissues to look at the contributions of the different cell types to that tissue. And so some of the different applications, in addition to what you've learned, is just resolving tissue composition, uncovering new cell types, understanding how cells change over development or time, as well as validating disease models. But of course, what's highlighted here is just a small sample of the different things that you can do with single cell data. However, the path to these insights from single cell data really depends on having an optimized sample prep workflow. So I'm just briefly going to talk about the different 10x Genomics single cell assays, mostly because each of these has distinct cell preparation requirements. So starting with the universal assays, these encompass things like 3' single cell gene expression and 5' single cell gene expression. By and large, you're going to be working with fresh or cryopreserved cells or nuclei isolated from fresh or frozen tissues. This is reverse transcription based, so it's whole transcriptome coverage. The next one is our flex assays, and so this one relies on a probe-based method for detecting gene expression information. So the probes cover the protein coding genome, or transcriptome rather, available for human and mouse samples. But part of the workflow or sample prep workflow for flex is to formaldehyde fix your cells, nuclei, and tissues before going into the workflow. Additionally, you can dissociate FFPE tissues and profile those with flex. And then lastly are the epichromatin assays. So this encompasses looking at accessible chromatin or attack data. And with these assays, you would want to isolate nuclei from fresh or frozen tissues or your cells, fresh cell suspensions or cryopreserved cell suspensions before going into the assay. And so as I mentioned, different assays are going to require different input materials. And so as I mentioned with the attack assays, you would have nuclei. But if you're just looking at gene expression, you have the option of either cells or nuclei. So really depending on what your experimental goals are, your sample type, that's where you would make your decision. But of course, when you want to look at other different modalities, you may need to work with cells. And so, for example, some of the ones that I wanted to highlight here are looking at cell surface protein measurements. So this relies on these DNA barcoded antibodies to stain your cells. And so in that case, you need to have an intact cell membrane to detect cell surface proteins. Additionally, a new enablement on flex gene expression, which of course requires that fixation, is intracellular protein profiling. And so in this case, you need to have an intact cell. So this would be a viable cell suspension that would go through a brief fixation and then staining with your intracellular protein panel, and then going into a harder fix, or a longer fix, excuse me. And then the last one that I wanted to highlight here, which also we strongly recommend using cells, is our VDJ receptor sequencing, so for TCR and BCR profiling. So that one you would also want to work with cells. So again, kind of determining your required cell input based off of your experimental goals can really help as you build out your sample prep plan. But I wanted to talk about a couple examples as it relates to cells and nuclei. And so this is a study that was published in Nature Medicine, came out of Stanford University, where the authors wanted to kind of understand the T-cell clonotype dynamics as it relates to anti-PD-1 treatment in basal cell carcinoma. And so in this case, they took their tumor biopsies from 11 different patients. So these were site-matched samples. I think total 24, 24 site-matched samples. And so these samples were dissociated fresh. And then once they were dissociated, the authors actually cryopreserved them, so that potentially is a batch point, before proceeding with the workflow. And so when they proceeded, after thawing those samples, they wanted to characterize those cells using FACS. And so they went ahead and stained with their FACS antibody panel, sorted out their different populations prior to running single cell, where they generated a gene expression library and a TCR library in this case. And so, and now that gives them the ability to kind of look at how those TCR clonotypes are changing related to that treatment. But in this case, of course, sorting out particular populations as well as that VDJ application, you would want to have intact cells for that type of an experiment. In another example, so this came out of McGill group for suicide studies, the authors wanted to better understand whether there were changes between or in major depressive disorder. And so in this example, the authors had collected samples post-mortem from their control cases, 17 control cases and 17 cases with major depressive disorder. These samples were flash frozen and then stored at -80 before going into a nuclei isolation, which in this case was a Dounce homogenization with a debris removal using OptiPrep gradient. But in this case, they were able to get kind of a broad perspective of the different cell types in the prefrontal cortex, so 26 different cell clusters that they identified, as well as identified some dysregulation in their excitatory neurons, as well as immature oligodendrocyte precursors. So again, in this case, when you're flash freezing tissue, you have the option to isolate nuclei from that sample. So the best advice is really to make a plan for your sample prep workflow. What is your sample type going to be? What do you have access to? What are your experimental goals? And determine how everything kind of comes together to what sort of assay you're going to use to run your single cell experiment. Another really important component is figuring out who's going to do what part of the workflow, and also very importantly, when a sample is going to be handed to a core to continue on with the single cell workflow. And I very frequently, especially for tissue types that haven't yet been, or it's a new tissue type for a user, I always recommend to run a sample prep pilot. So using a non-precious sample, they can run through the workflow steps, check the QC those samples, make sure it looks good in their hands before investing in a larger project with more precious samples. So we talked about this a bit in Mandovi's session, or presentation, but sample quality is really critical for single-cell sequencing. And so whether you're working with cells or nuclei, having an intact cell membrane or nuclear membrane is really key to kind of preventing high background in the assay. Additionally, when you have RNA that's leaking out of your cells and nuclei, that's just going to make the data much less informative for you. In terms of attack, so if you're preparing nuclei for attack, if you have a compromised nuclear membrane, a lot of times that chromatin structure, well, it can lose its structure if it gets too compromised. And so then again, the data isn't going to be particularly informative for you. All right, so getting started with sample preparation, we have a really amazing cell preparation guide available on our support website. It's going to cover kind of all the basics and beyond for sample prep. And it also has guidance for working with limited samples, which what we would consider to be less than 100,000 cells or nuclei. And so generally, the different steps in a sample preparation workflow, it kind of starts with sample collection. So maybe this is taking a biopsy. And from this point, maybe you work with groups that have easy access to getting to their lab space. They're going to run over with their sample on ice and dissociate it and proceed with the workflow. But sometimes there is a need to store samples in the meantime before they can be processed. And at this step, so it's this second gray box, if you're working with a tissue and maybe you need a few hours to perhaps a couple of days, you can consider some tissue storage solutions. So those tissues then get stored at 4 degrees before proceeding on with maybe a dissociation. You can also flash freeze the tissue if you're going to isolate nuclei from it. If you're going to run with flex, you can fix the tissue or freeze the tissue and fix the tissue later. So a lot of flexibility there. You then proceed on to your single cell or nuclei isolation. There's obviously many different approaches to this that Mandovi kind of addressed in her presentation. But then you also have an optional sample enrichment or depletion. And so this would also be a point where maybe if you checked your sample and saw that there were a lot of dead cells, you could consider a dead cell removal workflow. But I wanted to focus a bit more on the sample QC in the last few slides, just because this tends to be the point where groups will hand over their sample to a core to further process. And so generally, and we've heard this, ideally 90%, but I would say if it's 70% or above, that's probably gonna, especially for primary samples, this is a good metric to go by. When you start seeing your cell viability drop, that's usually an indicator that you're getting that sample degradation, and so the RNA is probably going to be leaking out of the cells and you're going to have high background. The other thing that you want to be considerate of is the cells and clumps, large pieces of debris. Again, if you're seeing a lot of clumps in clumping cells, you're not going to get single cell data. One thing that I wanted to point out is if you are working with a fixed cell sample, the viability will not be important here because they're all going to be dead. And I will also say that using a fluorescent counter can be really helpful in that case as well. When you're QCing nuclei suspensions, you'll see over on the right-hand side here, this is a 60x bright field of some nuclei. Typically, if you're QCing nuclei and visually, a 40x is probably going to be fine for you, but you just want to make sure that they're in good condition. So you'll see in panel A and B, we have nice resolved edges for those nuclei. You can even see some of the nucleolar structures. And as we continue on in C and D, we're starting to get blebbing, where it almost just looks like little bits of debris in panel D. The other thing that's important, and this is more for if you're going to be isolating nuclei from viable cell suspensions, so in the attack assay, for example, you want to make sure that you're getting good cell lysis, and so checking the viability of the sample. In this case, we want the nuclei to stain dead, so we want it to be about 95% dead when we're looking at nuclei suspensions. And again, fluorescent counters can be really great here using something like AOPI, especially if it's a debris-heavy nuclei suspension. The AOPI typically doesn't stain the debris in the sample and overcount that like trypan blue would. All right, so if the sample doesn't meet the criteria, and so this has been covered to a degree, there are various ways to clean up the sample, and these are also covered in the cell prep handbook. You can filter, maybe do some gentle washes, slow centrifugation, density gradients, and then more advanced methods for cleanup, which would include a dead cell removal. So we actually have a demonstrated protocol on our support site using the Miltenyi Max dead cell removal kit. As well as cell sorting, which can be used as well. And in that cell prep handbook, there's some nice guidance on sorter conditions for optimizing viability coming off of the sort. And so for additional resources in getting started, I've mentioned the support site a couple of times, but this is really a fantastic resource. It's got a lot of demonstrated protocols as well as technical notes, like a technical note on counting. Another thing that I wanted to point out is we have a publications webpage that you can link out to, and this is a fantastic resource. So you can sort this based off of your interest or tissue type, species, assay that was used, and it will then populate with different publications that used that particular sample type. And so this is a great way to see what's already been completed in the field, maybe learn about some strategies for sample prep and some nuances with that tissue type. So with that, I just want to thank everyone for your attention. Again, if sample prep, it's a very, I guess, nuanced process, and and there's a lot of resources for it. So if you ever have any questions, please do reach out to your local 10x Genomics team. We're always happy to chat with you, or our support team. We're always happy to look at cell suspensions and give you feedback on them. So with that, if there's time for questions, I'll take them.
Thank you, Dr. Cooper. So if anyone has a question for Dr. Cooper, 10x Genomics, or Dr. Chen from Takara, please do. Also, to quickly reply to the person who asked me why the cells were not fixed in space. I think it's you, correct? Yes. It's because I just remember, like in May 2023, the fixation kit from 10x Genomics did not exist, so we could not actually fix the cells. All good, okay. So thank you very much, everyone, for attending the session. I hope now you know how to actually do your sample prep for single-cell RNA-seq, and I hope you learn a lot during this session. Don't forget, for core managers and core directors, don't forget to mention that to your users. This is very important. Thank you.